Hi, everyone. Good afternoon and good morning. Thank you so much for joining us today. My name is Sharon Huang and the product manager at BGI Tech. So over the next half an hour, I'm about to share our comprehensive spatial analysis solutions with you. So just see how this Stereo-seq technology can be seamlessly applied to both fresh frozen and FFPE samples. Let's just dive right in. To understand why spatial transcriptomics is such a game changer, we have to look at how we have traditionally studied these tissues. For decades, we rely heavily on classical histological techniques. These are fantastic because they usually preserve the morphological context. So we have the in situ visualization of the original organ. On the other side of the spectrum, we had the genomic resolution with next generation sequencing. So the bulk RNA sequencing gives us the high-plex quantitative profiling, but conceptually, it's a bit like taking this logo brain and throwing it into a blender to make a smoothie, so we can get a accurate average expression of all the genes present, but we completely lose the track of the different cell clusterings and their structural organization. So we will see then came in the single cell RNA sequencing, which was a massive leap forward. It allows us to identify individual cells, like dismantling those Lego brain into individual distinct bricks. We know exactly what each brick is, but we still need to dissociate the tissue to get them, so we don't really have the critical and physical positions of those cell information. This is exactly where the spatial transcriptomics steps in as the ultimate bridge between those two worlds. So it allows us to perform the high throughput unbiased NGS while retaining the exact structural map of the tissue. So we can get the full molecular profile, and we know exactly where every single Lego brick belongs in the tissue architecture. So if you look at the timeline here, reaching this point didn't really happen overnight. So the evolution of spatial transcriptomics has moved through several distinct phases over the last few decades. In the early days, we had microdissection gene expression technologies. So we can use those technologies which allow us to isolate specific regions of interest for profiling. They were generally low throughput, labor intensive, and inevitably destroy the fine cellular content at that point. Following that, we also see the rise of the in situ hybridization and in situ sequencing technologies. So they are pretty powerful tools, incredibly powerful tools for targeted analysis and validation, offering the fantastic resolution as well. However, they come with a catch. You typically need to know exactly what gene targets you are looking for in advance. So they often struggle with the scaling up to unbiased whole transcriptome profiling due to the optical crowding and multiplexing constraints. The specific bottleneck is exactly why the field shifted towards in situ capturing technologies, and frankly, why we are all here today to see the Stereo-seq. The approach finally allows us to perform unbiased high throughput and the whole transcriptome profiling directly on intact tissue sections. So if you look forward towards the right side of the timeline, notice BGI's print. We introduced Stereo-seq around 2021, and it presented a massive paradigm shift by pushing those boundaries of the high resolution down to the nanoscale, and we have a pretty flexible on our chip size as well, so a different flexibility in the field of view. We also haven't stopped there. We introduced the Stereo-seq omics solution specifically designed for FFPE samples, reflecting our continuous commitment to unlocking those invaluable clinical archives. So how exactly does this technology achieve the performance? Let's just take a look under the hood at this core principles. During the manufacturing process, we would see there are millions of spots on the chip, and that's the reason why we have the high resolution of 500 nanometer. So the two biggest features here The first one, it would be the flexible chip size and ranging from standard 0.5 by 0.5 centimeter squares all the way up to a massive 13 by 13. This gives researchers the ability to map out the massive complex tissues, like the entire whole organ sections, in a single but continuous experiment. So we can just avoid the technical headaches of the stitching multiple small arrays together on a single chip. The second one is what I just mentioned. It has a pretty high resolution. The center-to-center distance between our capture spots on the chip is an incredibly tight 500 nanometers. So just put that into perspective. A typical mammalian cell is about 10 micrometers in diameter. This means that a single cell will be covered by approximately 400 of these individual capture spots. But as I just mentioned, how do we know those spots on the chip, and how can we know where exactly those millions of spots is located on the chip? During the process and before the tissue ever touches the chip, we utilize a technique called combinatorial probe anchor synthesis, or CPAS. You can see from the left-hand button figure. It basically provides the information about CID, which is the coordinate ID, which is just exactly the spatial information we have on the chip. So the CID information will be aligned to every single 500 nanometer spot on the array. The spatial map is therefore predefined with extreme accuracy. So after we capture the biological information on this chip, we usually have the readout. The readout is usually completed by our BGI's sequencing technology called DMB-seq. The downstream sequencing platform would use the rolling cycle amplification, RCA, to create the DNA nanoballs, DMB. So we would have high accurate, high throughput data generation that links back to our precise spatial map. So let's just look at how the Stereo-seq solution works for fresh frozen FF samples. For the fresh frozen samples, we usually have the capture strategy on their poly A tails. So the probe would just anchor uniformly across our pre-barcoded chip, just mentioned, feature with the poly T sequence. When we mount a fresh frozen tissue section onto the chip and apply a specific permeabilization step here, the cell membranes are just permeabilized and causing the release of the cells to their intracellular mRNA. Because the mature mRNA molecules naturally possess a poly A tail, they drop down and immediately specific captured by the poly T probes on the chip surface. The biochemical reaction here is the core of our spatial transcriptomics. It just directly and physically links the biological mRNA transcript to the specific CID barcode here. So let's just walk through the exact experimental workflow for fresh frozen samples in our lab step by step. First, we will usually begin with pre-quality control. So the embedded block, it will be sectioned. RNA would be extracted and tested for the integrity. Only high qualified samples are permitted to move forward to actual spatial chip. Next, we would have the step called permeabilization optimization here. Because biological tissues, they are usually highly heterogeneous. A brain doesn't really permeabilize the same way a liver does. So we must have different permeabilization timeframes on the practice sections. So we use the reverse transcription paired with the fluorescence imaging to locate the optimal timeframe that perfectly balances the maximum mRNA release with the preservation of the tissue's morphological structure. Once the optimal time is established, the actual experiment tissue sections are processed on the Stereo-seq chips. The tissue will be permeabilized and reverse transcribed. When we have the reverse transcription for the cDNA, the cDNA will be then cleaved from the chip, and then it will be purified, amplified, and then constructed into the sequencing libraries. And finally, these libraries are converted into DMBs and sequenced on our DMB platform. The final sequencing rate is highly constructed So it compromises the spatial CID, telling us where it came from, a molecular identifier, MID, to accurately count absolute molecular numbers and remove the PCR duplicates. The actual transcript sequence as well, telling us what gene it is, and of course the poly-DT track. Just to ensure we reach the transcription saturation for the standard 1 by 1 centimeter chip, utilizing the fresh frozen tissue, we typically recommend a sequencing data output of around 2G row rates. So this immense depth would be just ensuring we capture the full nonce complexity of the spatial transcriptome. Well, so for those fresh frozen samples, we need to spend a moment on something important. If you are sending those samples to our laboratories, some strict adherence to our preparation guidelines is required. So because the spatial transcriptomics technology is highly sensitive to the pre-analytical variables, the quality of the input directly dictates the quality of the output. So here are some critical requirements and common pitfalls to avoid the things that could be happened during the transportation or the pre-analytical process. The first thing you need to notice is the tissue size. The section must fit comfortably on the chip, so the size should be smaller than 80% of the Stereo-seq chip area, just to leave a safe margin for buffer handling and things like that. More importantly, we require an RNA integrating number of 4 or higher. So we would check out on that. The samples with significant degradation will just simply fail to release the intact poly-A tails, resulting in poor mRNA capture. So just do the quality control. We would just check if the tissue could go with a subsequent analysis. The second thing should be noticed here is the embedding process. The samples must be completely and carefully embedded in OCD compound. You must be extremely careful to avoid introducing any air bubbles during the process, because when the tissue is frozen, the bubbles become a bit brittle cavities that will cause the tissue to shatter, tear, or fall during the microtome sectioning, and it would just render the sample completely unusable for the continuous spatial analysis. So just also remember to clearly mark your desired cutting plan on the mood if you have a specific plan for your samples. The phrasing is also something need to be noticed here. The tissue should be snap frozen immediately after harvesting the RNA, just to stop the RNA degradation. We strongly recommend using dry ice or isopentine chilled with the liquid nitrogen, depending on your sample type. And for the shipping process, we would recommend the dry ice and the freeze thaw cycles should be avoided at all costs. Even a single partial thaw cycle can probably destroy your sample during the transportation. And also something to mention here, the special tissues, for example, those highly challenging tissues, the classified bone or the high-fat, high-RNA tissues like pancreas may require highly specialized pre-handling protocols, but just please consult our technical team before you even start your collection process. So just for the precise parameters and everything on board, please thoroughly review our detailed sample submission menu before initiating your collection. Next, just moving on to a major breakthrough for clinical research, our Stereo-seq OMI solution for Formalin-Fixed Paraffin-Embedded samples, the FFPE samples. So this could just help us to unlock the massive archives of the historical clinical cohorts, of course. The fundamental architecture of the chip remains identical to what we just discussed for fresh frozen tissues. We still rely on our incredibly dense DMB array and the CPAT process to assign a unique spatial barcode to every 500 nanometer spot. But we do have a different capture chemistry here. As many of you may know that the FFPE processing heavily cross-links and severely degrades the RNA. So that means we cannot rely on the intact poly-A tails. Therefore, for the FFPE chips, we use another thing called random probes here. The random priming strategy allows us to just effectively catch those highly fragmented RNA molecules directly from the cross-linked tissues and ensures we still get the robust high-quality transcriptome data despite the challenging sample type. So we can even get the total RNA profiling, something from a long non-coding RNA, and we can detect the microbiome at certain level. The experimental workflow for FFPE is pretty similar to what we have for fetch-frozen protocol. The downstream steps for the library construction and sequencing remain largely the same, but here are some critical differences in the upstream processing. So first thing is we do have some the paraffinization and the cross-linking steps to just expose the nucleases, but the most time saver and biggest operational difference here is the fixed permeabilization time. Unlike fresh frozen samples, FFPE tissues don't require the permeabilization optimization because the tissue matrix is already heavily modified by the formalin fixation. So we would use a single fixed time for all the FFPE samples. It just makes the workflow highly standardized and significantly reduce the hands-on optimization time. So even though we use the random probes instead of the poly-T for FFPE samples, the final sequence library structure is pretty similar as well. It still contains the vital spatial CID, the molecular MID, and the transcript sequence. Just like with our fetch-frozen protocol, we typically recommend a sequencing data output at around 2.0 rates for a standard 1x1 centimeter chip, just to ensure you can reach the full transcriptomic saturation. So FFPE sample do have some other requirements as well. Here's some core requirements. So as always, the tissue must fit comfortably within the target capture area, remaining smaller than 80% of the chip active surface. And if you're sending the paraffin sections, they must be flat and intact. The tissue with folds, wrinkles, tears, or significant scratches are unacceptable. Any physical distortion on the side will just on the section will just directly translate into the distorted spatial data and compromise the structural map. We also do the RNA quality control here, but we don't really use ring value. But here we have DV200 metric, which is the percentage of the RNA fragments longer than 200 nucleotides. So a higher DV200 value is really critical for ensuring sufficient spatial library complexity. We just have a requirement for the fixation standardization as well. So the FFPE samples should be within a controlled fixation process, typically using 10% neutral buffered formalin for 18 to 40 hours. So the prolonged overfixation could just limit the RNA extractability and will compromise your data as well. For the packaging and shipping, we accept both FFPE blocks and sections. For the sections, we recommend 2 containers options to prevent the physical damage. The option 1 is a 50 milliliter centrifuge tube with 1 section per tube. Option 2 would be a paraffin-lined Petri dish, which can fit single or the serial continuous sections. So unlike FF samples, FFPE samples do not require the dry ice, but we do really require they are shipped with the standard ice packs and with desiccant. Again, just please thoroughly review our sample submission menu and consult our technical team before sending your sample cohorts. Just a quick summary on those 2 solutions here. The Stereo-seq solution is designed for the frozen samples. You would capture the mRNA by using the poly-T sequence capturing method. And the Stereo-seq only solution here is designed for the Formalin-Fixed Paraffin-Embedded samples. They would capture the total RNA within the tissue with the help of random probes. They are both available for ssDNA or HE staining, and they have two standard chip size, like 0.5 and S1 chip. And here I would like to address that we do offer more different chip size, but they just need to you just need to email the technical team to do the further consultation. And both solutions, they are species agnostic and have the same recommended sequencing throughput for the two different standard chip size. So we have covered the lab workflow and sequenced the samples. We have a massive amount of spatial transcriptomic data here. But before we dive into the analysis pipelines, it's critical to understand the concept of being in Stereo-seq data as well. You can, of course, use our softwares or the softwares in the community to do the cell segmentation to get the true single cell data. But we do have the idea of being here. So the distance between our capture spots, as I just mentioned, is only 500 nanometers. If we analyze the data spot by spot, that's what we call being one. We are looking at the true subcellular level. While this resolution is incredibly powerful for certain studies, it's often too granular and computationally heavy when you are trying to identify the border tissue domains or major cellular clusters. So to solve this, our software aggregates those 500 nanometer spots into a functional grid called bins. For example, the bin 20 aggregates a 20 by 20 grid of the spots. Because each spot is 500 nanometers, bin 20 covers a 10 by 10 micrometer area. This specific bin size is highly utilized in research because it roughly approximates the size of a single standard mammalian cell. If you zoom out further, bin 50 also works, or bin 50 or even bin 100 are commonly used to quickly evaluate larger morphological structures, the broad tissue domains, or just to get a bird's eye view of the tumor microenvironments. We also do offer things like bin 200 as well, just depending on your requirements and needs for the analysis. We just have a pretty high flexibility here, and you could just do all the analysis yourself. We do offer the in-house bioinformatics softwares here to provide the biological insights. We have a pretty complex ecosystem built around a standard closed loop workflow consisting of the three different phases. The phase one will be more about image processing. So we start with the physical tissue images like your fluorescent or the bright field scans. This SAW here, you could use it to handle the heavy lifting of the image restoration, segmentation, and background removal. But if a sample has complex morphology, researchers can use our visualization tool called Stereo-map here to manually adjust and ensure the spatial coordinates are perfectly aligned. This could just create our accurate geographical map of the tissue. The phase two would be mainly done by SAW. It takes the raw sequencing rates and maps them into the spatial coordinates from phase one. The SAW calculates the key metrics like the MID count, presenting the true unique RNA molecules and information about the exon count and cell ID, things like that. So the SAW output here is just the spatial gene expression metric after a whole pre-processing of the raw data. The final phase here would be visualization and the additional analysis. We could use the Stereo-map to overlay the gene expression metric directly onto the tissue image. You can instantly see the spatial expression patterns. For example, a gene that is highly expressed at the tumor margin, but completely absent in the necrotic core. So from here, the data matrix is fully compatible with the other diverse community developed tools like ScanPy as well, just to do more about the downstream mining. We do offer the end-to-end bioinformatics services. The offerings are structured into two main tiers, standard and advanced analysis. The standard analysis is the fundamental workflow. It essentially answers the question, is the data high quality, or what does the basic landscape look like? So as you can see from the left side of the chart, it takes the three essential inputs, the reference genome, the FASTQ sequencing rates, and the tissue mask containing the CID information. It runs through the core SAW pipeline to generate the crucial spatial expression metric. So the standard output gives you the baseline of the tissue, basically. The advanced analysis on the right side of the chart is where we answer the question if there are some specific biological story behind the data. This tier is highly customizable based on your research goals. Crucially, it allows you to integrate your own existing single-cell RNA sequencing data or the public data sets containing single-cell RNA information that corresponds to your tissue, just to improve the accuracy of the cell type annotation in your spatial map. So we can also use the lasso tool in the stereo map, manually isolate the very specific regions of interest for targeted analysis. But also just before getting the information, you would just also get everything from a standard analysis as well. Just to get you a bit more information about the advanced analysis here. We also offered some advanced modules. For example, if you are studying the oncology, you might utilize the cell-cell interaction modules to map exactly how tumor cells are communicating with the immune cells at spatial boundaries. Or if you are studying the developmental biology, you might utilize the pseudo-time analysis module to reconstruct the cellular differentiation trajectories while preserving their actual spatial distribution in a developing embryo. So where is this technology actually being applied? The applications are extremely wide, but we can see massive adoption and breakthroughs in several key fields. The developmental biology, researchers are mapping those embryos genesis and organogenesis in both time and space, tracking how the gene expression drives the structural formation. And it could be applied in pathology as well. It could be heavily used to dissect the tumor microenvironment, specifically identifying the immune cell infiltration at tumor boundaries and discovering the spatial biomarkers for those disease progression as well. We also applied it to the neuroscience, where you can just map different cell types across different regions and see if those gene expression could be different to the healthy or the unhealthy tissues. We also apply that to regenerative biology, just to identify those regenerative cell types and different signaling nodes. And we can just do that across species as well, just do a comparison between those animals. You can get more idea and more biological insights from the sample. The scientific validation speaks for itself. So as the end of the last month, Stereo-seq has enabled researchers worldwide to push over 500 research papers. Over 200 papers have generated the new Stereo-seq data themselves, and some of them also cover stories in top journals such as the Cell, Nature, and Science. The number is still increasing really rapidly, and you could probably see some new papers coming out recently as well. And I like to highlight a few examples here. Let's start with the developmental biology. The main objective in this paper was to digitally reconstruct the full mouse embryos during the early organogenesis at true single-cell resolution. To achieve this, the researchers sourced the complete wild-type mouse embryos at developmental stages from 3 different developmental stages here. They utilized Stereo-seq to comprehensively map the spatial transcriptomes, generating over 200 high-quality coronal sections that were successfully aligned to a 3D model here. So we mentioned the beam concept before, but in this study, they don't really rely on the traditional fixed grids like beam 20 or something. They took it a step further to achieve the true single cell segmentation directly on the ssDNA stain images. This is done by the cell beam function from StereoMap and cell pose from the community. The figure here demonstrates the core features of their SEU 3D platform on the left-hand side, highlighting the interactive 3D morphological modeling. So if you see the sagittal sections on the right-hand side, you can just see the dynamic spatial localization of the specific cell types across the ectoderm, mesoderm, and endoderm over time. So this paper automatically provides a powerful high-resolution spatial atlas just to make sure that we can understand the full embryos completely. And here is more about neuroscience and metabolic disorders. The slide presents a study investigating the molecular and spatial responses of the primate hypothalamus to diabetes. This is a perfect example of a multi-omics approach. The team employed 2 main methods. First, they used the single nuclease RNA sequencing. It's from our DNB ELAB C4 platform to profile the transcriptomes. I don't know if you joined the single cell session before. It's more just about our DNB ELAB C series. And we do have additional webinar session, I think next month in April as well. And secondly, they also use the Stereo-seq to map the cells back to their exact spatial coordinates in the tissue. So by using those 10 micrometer thick sections from the adult male macacus, they align the bypass traditional beams and utilize a tool called Spatial, also developed by BGI, for the automatic cell segmentation based on the ssDNA staining. The figures here just demonstrate the power of the dual omics strategy. The large spatial map on the right, you can see a very complex distribution of virus cell types across the different hypothematic nuclei. But the most crucial pathological finding is in the smaller line graphs in the upper middle. In the diabetic state, the data could just show that the activated microglia physically migrate away from the epidermal lining and they just dip into the parenchyma. They conclude that there's something happening within the cells. So if we had only used the standard single cell sequence here, we would probably just know the microglia were activated, but we would have absolutely no idea where they are migrating to. The spatial atlas pinpoints the importance of the region-specific vulnerabilities and offers the highly valuable new targets for treating metabolic disease here. This is another clinical application in oncology by Stereo-seq only. The study aimed to evaluate how the combination therapy can overcome the immunosuppressive tumor microenvironment of the pancreatic cancer or PDAC. To uncover the underlying mechanisms of the drug efficacy, they applied both bulk RNA sequencing and our Stereo-seq spatial transcriptomics technology here. And crucially, they applied the FFPE tumor samples from their mouse models. They collect the FFPE samples representing 4 different treatment groups, the control, FOLFOX, L19, IL2, and the combination therapy. They're assembled into a single tissue microarray, TMA. 1 5 micrometer section of the TMA was sequenced and analyzed. you can see they analyzed this at being 100 resolution. So as we just discussed earlier, this is perfect micro level being sized to evaluate the broader tumor microenvironment. And the most critical findings are in the panel CD on the right-hand side. They just visually demonstrate that the combination therapy successfully remodels a cold tumor into a flat hot state. You can see that the massive spatial infiltration of the CD8 positive T cells and NK cells, the antigen presenting cells, directly into the tumor core. And ultimately, the targeted spatial strategy paints a promising vision for improving standard chemotherapy and plus the immunotherapy outcomes in PDAC patients. So this is another application in neuroscience, so this time focusing on the neurodegenerative generation and actual clinical translation here. The study maps the molecular and cellular changes in the Alzheimer's hippocampus to uncover the disease mechanisms and specially defined clinical biomarkers. To achieve this, the team designed a masterclass multimodal study. They combined the Stereo-seq for spatial mapping with DMB-ELAP-C4, the single nuclear RNA sequencing to provide the specific cellular subpopulations. Then they utilized the IHC-IF staining to validate the A-beta pathology. And finally, they also used the clinical cohort to verify the newly discovered biomarkers there. Just for the spatial mapping, they applied highly precious post-mortem frozen hippocampus tissues from 16 individuals. They 8 with moderate to advanced AD and 8 healthy controls, sequencing 32 tissue sections in total. The figures on the slide perfectly just says that these technical capabilities here. So because from the bottom panel, you can see that how it could map the complex and anatomical the subregions of the human hippocampus based purely on the spatial gene expression profiles here. And the bottom right map the precise spatial distribution of the 18 distinct cell types between the control and AD samples at a single cell level. And the top right embeds the exact coordinates of the amyloid beta plex directly into the spatial cellular map here. So this study provides a comprehensive atlas here, identifying specific microenvironmental changes driven by those amyloid beta plex and successfully translated those findings into the tangible liquid biopsy biomarkers for the AD diagnosis. The final case study here, published last year, is a fascinating dive into the regenerative biology here. It asks a profound question that why can some mammals regenerate lost appendages while the others simply form scars? And more importantly, can we reactivate the ability here? So to answer this, researchers applied a comprehensive multi-omics approach as well. First, the bulk and single-cell RNA sequencing pinpointed the wound-induced fibroblasts, or the WIFs. While doing that, we could get the primary cellular drivers of the regeneration, but to understand exactly where and how those cells act, they needed the spatial context here. So the team applied a Stereo-seq on the frozen ear pinna sections from rabbits, which can regenerate, and mice, which cannot at 10, 5, and 10 days post-injury. As demonstrated in the spatial maps here on the center, the construct between the species is pretty stark. Figure on the left hand shows a highly organized pre-regenerative blastema forming in rabbits, but conversely, a non-regenerative inflammation-heavy tissue repair environment is shown in mice. So the top right figure also further confirms the critical morphological genetic genes here. They are expressed only in rapid spatial niche, but not in mice. Just by integrating those spatial findings with the epigenetic data, they then discover the exact mechanisms. A disabled enhancer marker just blocks the production, the retinoic acid production in mice. So that's why they could get the whole story for the paper. By genetically or chemically reactivating the switch, they could restore the tissue regeneration in the mice. So with the help of the Stereo-seq information here, they could just link all the molecular parts and to the protein validation part, and offering a groundbreaking vision for the future of human regenerative medicine. Just to summarize our technical capabilities before we move to your questions, I would like to leave you with the 4 core pillars of the BGI spatial solutions here. So we have a pretty high resolution here, true nanoscale subcellular resolution at 500 nanometers, ensuring that you never miss a single cell, and we pair that high resolution with a unmatched panoramic field of view, scaling all the way up to 13 by 30 centimeters. We also provide the complete unbiased whole transcriptome capture, the species agnostic. You don't need to choose your targets in advance and get the whole profiling information. And we offer the high optimized standardized workflow, just for both fresh frozen and challenging FFPE tissues, empowering the research across a wide range of species and sample types. Just want to say thank you so much for your time and attention today. We are excited to see how could you use the Stereo-seq to map the frontiers of the life in your own research. These are some information about our official websites. You can just scan the QR code or just call the phone number and visit our website with the link as well.